Hello, students. I just wanted to give you a little bit more information about the bean experiment. This is in lab number 2, and I often get many questions about this. So to start out, I have up here on the Moodle site the bean experiment and scientific method information. So this contains the background information for this lab. I know there's a lot to read here, but it takes you through the steps of the scientific method, um, one by one in describing the different methods. Now, the scientific method is an important way for us to study the world because it allows us to form a hypothesis, an idea, and test that hypothesis, and collect data and make some logical conclusions. So you can use this in science. You could also, and you probably do, use this in everyday life. In an experiment, when you design it, you'll have a few different variables, and they're described in this part of the page. Independent variables are what you alter in your experiment. Say you want to test the effect of fertilizer on plant growth. Fertilizer would be your independent variable, the thing you're testing. Medical research. Right now, people are testing new vaccines for the coronavirus. And so they'll have a placebo group and a test group that gets the new vaccine. And that vaccine is the independent variable. Its effectiveness is measured as the dependent variable. And then controlled variables are things that are kept constant between the two groups, between the independent variable group and the control group, or the experimental group and the control group. So things like, if you were going to do the fertilizer experiment with plants, you would have plants given water as your control group, plants given fertilizer as your experimental group, and you would keep the species the same. You would give them the same amount of sunlight and give them the same amount of soil and that type of thing. Those are controlled variables. This is a flow chart showing how the scientific method isn't always just a straight line. Sometimes when we do research, we come up with unusual conclusions or things that lead to more questions, results that lead to more questions, and we can go back and construct another hypothesis in that case. It's one of the exciting things about being a scientist. When you're wrong, it just leads to more science. And down here, we start the bean experiment. So there are two parts to this lab. The first part is designing your experiment, and the second part is doing it and writing up a report. So here we're looking at kind of the overview of what we're going to be doing in this introduction. You're going to be taking 30 beans, splitting them up between 6 plastic sandwich bags. So you'll have 5 beans, 5 beans, 5 beans. And that will be your replicates for your experimental group, and then 5 beans, 5 beans, 5 beans as replicates for your control group. Now, having these replicates ensures that if something goes wrong in one of those bags, it doesn't affect your whole experiment. So it's something that scientists do to ensure that the results will be consistent, that we don't have to do everything over and over. So you're going to be germinating these beans. Make sure to get dry beans from a grocery store, not in the can, but something dry. If you have bean seeds from a garden, those work too. But you can get like regular soup beans or navy beans. Anything really works. I want you to consider how these beans sprout. How do plants grow? What do plants need to grow? And then choose one variable, one thing that you want to alter to see if it makes a difference. So sometimes students will decide to test light, and so they think plants need light to sprout and grow. So they'll put 3 experimental bags in the light and keep 3 experimental bags in the dark and collect data on how many sprout as to see if there's a difference. Sometimes people will add fertilizer to one and just the wet paper towel to the other, and that can test whether fertilizer has an effect. Or you might want to put vinegar in one bag to see if acids have an effect on your beans. It's up to you what you choose to do. If you want to run any ideas by me, I'm more than happy to give you feedback about that. So here you're just thinking about how you're going to do your experiment. The procedure that's listed here is a general procedure on how to get this set up to start. And it's kind of the procedure you would use for your control group. You could just use this straight procedure and then alter one variable for your experimental group. So the first thing you need to do is to think about this and fill out this assignment. This is a separate assignment on the Moodle page if you prefer that. It's just a document, and this lets you think through what your variables are going to be and what your groups will be. This is due on November 9th. So once you turn that in, you can wait for feedback from me. And if you really want feedback, definitely send me an email and give me a heads up that you want to know immediately. Then you can fill out your data. Set up your experiment. You're actually going to run this experiment at your house. Now, I have a general data table here with days and the number of beans germinated. You're welcome to use this, or you can modify it depending on your experiment. So you could just put day 1, 2, 3, 4, and then count the number of beans that have germinated. So start with day 1, and you'll have 0 here. Zeros are okay. They're data. So don't neglect that. If you have 1 germinate here, write down a 1. If you have another one germinate here, this is important, put a 2. You want to be additive here. Not the number of new beans that germinated, but the total number of beans that germinated. If that is the data you are collecting, I recommend that because it's the easiest way to see if there's a difference here. You can also alternatively record how big your germination is, like how long are the roots or how tall is your sprout. Okay, I'm just going down here because I want to point out that I have a table for your experimental group and a separate table for the control group. So you could do collective data for all 3 bags on one table if you'd like, or you could do separate tables for each bag. It's up to you, and I know you may have questions about this. It's very open-ended, and you're welcome to send me an email with any questions you have. We could even set up a Zoom if you want to go over things, bounce ideas off of me. Also, if that's even a little bit too much for you, we can do instant messaging and set up a time to do that, or a phone call. Happy to do that. Okay, so you'll collect that data and give yourself about 7 to 10 days of data collection. If you want to go a little longer, that's fine too. But usually if you're doing germination rates and how many germinate, they'll all have germinated by about day 10, all that will germinate. This describes the mini lab report that you will hand in. Now, I do see that I want to update this a little bit and add some details here about exactly what I'm looking for. But your lab report will have an introduction section, a methods, results, and a conclusion section. One thing I'll want in your lab report is a graph, and there are some different ways to do that. ChartGo can help. It's a free online graphing tool, which is really easy to use. I have a tutorial on our lab page. So this is what we're looking at for lab number 2, and I look forward to hearing from you. Let me know if you have any questions. And by the way, I will often give you a week or so to do assignments, sometimes more than that. I like to open them up so there's plenty of time. So sometimes you'll see that I post things on the weekends. This class being asynchronous can happen for you anytime that works for you. So some students are doing all their work on the weekends and working during the week. Some are doing classes during the week. I'm kind of squeezing in my work whenever I can, so I know what that's like. Let me know if you need anything, and I'm happy to help.